Good morning to all. So after a brief introduction on introduction of biochemistry, now we'll properly go into the biochemistry topics. So for anything we have to start from basic level. So first we'll learn what is a cell. What is a cell? Now the cell details and everything you have, like everything you know about it, nothing so much new of it, but we'll have a look in medical perspective. Now, the cellular organization of an animal or any human being is that the basic thing is any cell organelle. First, a cell organelle begins. The cell organelles, the communication or the collection of cell organelles will form a cell. So, cell is a basic and functional unit of the life. Many cells club together and they form tissues. When tissues organize with each other and form a structure that has got a particular function, then you call it as an organ. So, the subcellular organs will be there. These all the organelles will combine to form a cell. Like multiple cells collectively they form a tissues. When tissues come and organize itself and they form a particular function, then you call it as an organ. Now, a organ system involves, it is a collection of all these organs like brain, kidneys, liver, um, all these digestive systems, so all these things, muscular system, skeletal system. So in that way, all these organ systems collectively they form an organism. So all these things collectively form an organism. So this is basically the evolution of a cell, from cell organelle to a cell to tissues to organ system to a complete organ. So this is the evolution of an organ. Now, the cell is a basic and the smallest functional and structural unit of an organism, as we already know. It is an independent entity and it can metabolize on its own. The importance of the cell is that it itself will act as a separate entity. It does not require anybody's help because it can get its own nutrients, it can provide its own energy because it is able to synthesize its own energy, and the most important part is that it can replicate itself. All these things can be done by a single cell. A single cell can do all these things. So it knows what to do for its survival and for its proliferation. So it has got its own nutrients, it has got its own metabolism, it provides energy, it derives energy from the metabolism. If anything is excess, it stores it in the cell, and most important is that even though it perishes, it will continue like that in the form of its progeny by means that it has got an important property of replication. Now, innumerable chemical reactions occur in a cell. So, these innumerable chemical reactions, in the last class what we were talking about, the metabolical processes, all these things, these are the innumerable chemical reactions occurring in a cell which contribute to life and procreation of a cell. Now, And as you know, there are two types of cells because we have evolved from prokaryotes to eukaryotes. So there are two types of cells called as prokaryotes and eukaryotes. In a developed individual, they have eukaryotes, and in primitive are the prokaryotes. The main difference between both of them exists is the presence of a cell membrane. The presence of a proper cell membrane differentiates both of them. So though they have a cell membrane, they usually have a cell wall, and inside that cell membrane we call it as a plasma membrane. We specifically do not use the word cell membrane. We basically use a term called as plasma membrane here. So a cell has a cell wall. Few prokaryotes it can have a cell walls, and it has got a nuclear capsule. The capsule is important in prokaryotes because most of the prokaryotes are pathogenic in nature. So they do present in some parts of the other, so they have to protect themselves. So for that they have a capsule that is completely resistant to different types of circumstances, so that cell doesn't get damaged. Now, coming to eukaryotes, the eukaryotes have a clear and well-defined cell membrane will be there. That is number one. Number two, inside they'll have a cytoplasmic membrane will be there, and inside that there are many organelles that are present that help in functioning of the particular cell. Whereas, and one more important thing is that its nucleus is present in the center, and it is covered by a membrane called as nuclear membrane. So it is covered by a nuclear membrane. That is very much important. That will be differentiating a prokaryotic and an eukaryotic cell. Here, the nucleus or the genetic material is present freely. It is not surrounded by any specific enzyme. Cell organelles are present very minimal in a prokaryotic cell, but they will be having some amount of ribosomes. All these are there. And flagellum is very importantly present because it helps in mobility. Now, in a eukaryotic cell, there is a nucleus is there that has a nuclear material. Then in the center, nucleolus is there. Then after that, multiple cell organelles are there. If we start with, there is endoplasmic reticulum. They have Golgi apparatus. They have cytoplasm in which all the cell organelles are present. There will be mitochondria. There are lysosomes, peroxisomes, and most important are the cytoskeleton. This is called a cytoskeleton. The fibrils that will attain the shape of the cell and prevent the cell from getting collapsed. And most important is that each cell organelle has got its own membrane. It is not that it is floating on the in the cytoplasm, but it has got its own cell membrane. That is a very important feature of all the cell organelles that are present in eukaryotic cell. Now, so cell organelles, there are different types of membrane enclosed bodies in a cell performing unique functions. So each cell organelle has got a unique function. An understanding of functioning of cell is required to understand the diagnostic enzymes, metabolic disease processes, hormone-specific diseases, and treatments. So for that reason, you have to understand the functioning of a cell to have a better idea of all these things. So that is why you have to learn the structure and functions of a cell. Now, to start with, we'll be starting with the very important part of a cell, that is the nucleus. The nucleus, it is one of the, I won't say it is the largest cell organelles, but it is one of the largest cell organelles, Most important and the classical feature of nucleus, like human bodies, how brain is so important, in that way to a cell, nucleus is that much important. So because the nucleus contains genetic material which is tightly packed as chromatin. It is tightly packed as chromatin, and you all know how many chromosomes we have. We have 46 chromosomes. Now, the genetic material that is tightly packed in the chromatin, and in the center there is one more substance called as nucleolus. So, this nucleolus is nothing but a center core in which the transcription process usually takes place inside the nucleolus. The transcription process usually takes place in that. The important feature of another important feature is that it has got a nuclear membrane. It has got a nuclear membrane is present, and that is not permeable to any of the substance. That is completely the transport of substances is very much limited or guarded through the nuclear pores. Now, one more thing, and the very important functions of the nucleus is they help in cell growth, mainly everything like how brain controls all parts of the body. In that way, nucleus helps in controlling all the functions of a cell. So, the nucleus mainly responsible for the cell growth. It is required for the protein synthesis. If a particular protein or particular enzyme is required by a cell, so what will happen? It will give stimulus to the nucleus. It will it will send its message to the nucleus, and the nucleus will be synthesizing that particular protein. And if you remember, there is a process called as transcription and then translation, in which protein is formed. First, via the transcription, there is formation of mRNA. That mRNA will come out of the nuclear membrane and it will enter into the cytosol. Then translation of that mRNA takes place, and that will result in the formation of a particular protein. So all these things will occur, and very important thing is that it helps in cell division. So in cell division, so all the properties of the cell, propagation of the genetic material is very important. If there is no propagation of genetic material, the cell will stop at that level. Growth will be stopped. If anything has to grow, means division has to take place. So the nucleolus cell division will take place in the nucleus. That is a very important thing. So the nucleus is the organ. The nucleus is an organelle that controls all parts, all organelles of a cell. It contains genetic material. It helps in the cell growth, protein synthesis, and also in the cell division. Next is endoplasmic reticulum. This endoplasmic reticulum is an entity that is present in continuous with the nuclear membrane. So it is so whatever that has come out of it, they can get incorporated into the endoplasmic reticulum. So in that way, endoplasmic reticulum is in continuous with the nuclear core. So as I told, the protein synthesis takes place on the ribosomes, and these ribosomes are as protein synthesis takes place in the endoplasmic reticulum, these ribosomes are plugged, or they are embedded in the endoplasmic reticulum, and that is why we call it as rough endoplasmic reticulum. It looks very rough when ribosomes get embedded on that. So these are the sites where protein synthesis occur. As I already told you, transcription process takes place in the in the nucleus, and the mRNA will come out of that. That mRNA is transferred with the help of ribosomes on rough endoplasmic reticulum. Then smooth endoplasmic reticulum. Some part of the endoplasmic reticulum is devoid of the ribosomes, which we call it as smooth endoplasmic reticulum. This is the site where the lipids and steroid hormones are usually synthesized, because the body will be needing fatty acids. They are synthesized, and also the steroid hormones are synthesized in that region. Next is the Golgi apparatus. Now a protein has formed from the rough endoplasmic reticulum. From the rough endoplasmic reticulum, a protein is formed. Now, when a raw material is formed, it is not completely suitable. When you get a piece of cloth from a factory or something, is it suitable to wear? No, it is not suitable to wear. So what we do, we'll send it to a tailor, and the tailor will fix it. The tailor will fix it, or the tailor will adjust in such a way that that becomes functional. Sometimes we get dresses from we get the fitting is not proper. So what do we get a dress, but the fitting is not proper, so we will give it to the tailor, and he'll make adjustment, and he will see that it fits us exactly. So initially when you have brought it from the stores, that was not functional, that you won't be the purpose of the dress is that you have to wear it, but you are not able to wear it. Now when you are sending to a tailor, he is making so many adjustments that now you are able to use that particular dress. In the same way, the protein that is formed from the endoplasmic reticulum with the help of translation process. See, the protein is not formed by the endoplasmic reticulum. It is formed on the mRNA with the help of the ribosomes. Because ribosomes are implanted on the endoplasmic reticulum, the process occurs in that way. So, the protein that is formed after the translation process is not that very suitable to perform the function. So it has to go to a tailor so that it gets adjusted. So the tailor in our body is the Golgi apparatus. Now these Golgi apparatus, and sometimes what will happen, the proteins that are synthesized, they are not utilized in the cell. They have to go to some other part of the body. So for that, transportation should be available. So what will happen, that unmodified protein will enter into the cis phase of the Golgi apparatus. So Golgi apparatus have got a cis phase and a trans phase. Cis is the one where the protein enters into that. Then passing through this membranous structure, you can see how the Golgi apparatus structure is. So passing through the membranes of the structure, that protein will undergo modification. So what all modification usually the protein undergoes are usually hydroxylation of some of the chains. The protein will get hydroxylated, or some tails that are present on either of the ends, those tails are removed. Sometimes some more bonds are added, sometimes those bonds are being removed. So like that, multiple things will happen, and finally, from the trans phase, a protein, the functioning protein that is now able to function perfectly will come out. If the protein has to be utilized by this cell only, then it will directly come out of the Golgi. But if this protein that is formed is not utilized by the cell, but it has to go and act on some other organelle, it will pinch it off. So these Golgi apparatus will pinch it, it will collect it in a small vesicle, and that vesicle is pinched out of the Golgi apparatus, and it will come like that. Slowly this will come out of the cell membrane. It will travel through the cytoplasm. It will come out of the cell membrane and it will go to its target organ. So the Golgi apparatus is responsible for transportation, modifying and packing of proteins and also lipids. So that is the function of Golgi apparatus. Next are lysosomes. Now, when we are saying that the cell is a complete, it is a unit by itself, then obviously if our house is unit by ourselves. So our house requires a dustbin is also required. Where you can't say it as a dustbin, but a place where the bad things or the dirt or anything, it has to be sterilized, so sort of sterilizer or something. So a lysosome is a subcellular, it is an organelle that will act as waste disposal system. One extra Y is there. So it acts as a waste disposal system to that particular cell. So the lysosomes, inside the lysosomes, there are various hydrolytical enzymes. There are various hydrolytical enzymes. These hydrolytical enzymes will come. These hydrolytical enzymes are released, and whenever any foreign particle enters into the cell, the lysosomes will take hold of it. The lysosomes will, via the phagocytosis, they'll engulf that foreign substance, and by means of hydrolytical enzymes, everything gets destroyed. So the lysosomes are the ones that will help in waste disposal of a cell. Next are peroxisomes. The peroxisomes also has a lipid bilayer, and it has got a crystalline center core. The important thing about the peroxisomes is that these are involved in oxidation reactions. The peroxisomes are involved in oxidation reactions. So what will happen, because these are involved in oxidation reactions, there are chances of more and more production of hydrogen peroxide. So what will happen, how the lysosomes help in destroying? The peroxisomes also have a role in destroying, but the capacity by the peroxisomes to destroy the bacteria is very high when compared to the lysosomes. So they generate hydrogen peroxide. This hydrogen peroxide is a very strong sort of a free radical. It is not a free radical, but it is a very strong one that can destroy the substances. So when they get engulfed, the hydrogen peroxide is released, and that will damage that particular bacteria and kill the bacteria. And one more function of peroxisomes is oxidation of long chain fatty acids. The long chain fatty acids are oxidized and they are broken down into smaller fatty acid chains so that these can be happily acted upon by the other enzymes. So very long chain fatty acids cannot be broken down by the cytoplasmic enzyme. So the breaking down of those very long chain fatty acids will take place in the peroxisome. So that is a function of peroxisome. Next, mitochondria. Mitochondria, I need not tell you about mitochondria because mitochondria, the first word that comes to your brain when I say a mitochondria is the thing that you are learning from your 6th class, mitochondria. It is the powerhouse of a cell. So that phrase is like every book, if it starts with cell in mitochondria, the first line in any book will be mitochondria is powerhouse of a cell. Now, why do we call it as a powerhouse of a cell? Because this is an organelle where ATP is generated or ATP is synthesized. And the structure of this is also a little bit different when compared to all other cell organelles. Like we have two membranes, here two membranes are there, partition is there, like cytoplasm thing, you have something called as matrix here. And important is that it has got its own DNA. The DNA is not as efficient as that of the cell DNA, but mitochondria has got its own DNA. So, looking into the structure of the mitochondria has got two membranes. It is called as an outer mitochondrial membrane and inner mitochondrial membrane. The inner mitochondrial membrane is wavy like this. It is wavy like that, and in between the membranes there are some threads which are called as crystals. These are called as intercrystal spaces, and in between and inside the inner mitochondrial membrane, the fluid-like thing that is present is called as a matrix, or we call it as a cytoplasm. In that way, it is called as matrix, and the space between the outer membrane and inner membrane is called as intermitochondrial space. It is called as intermitochondrial space. Now, once we look into the mitochondrial membrane, the matrix is embedded with the enzymes that are required for biological oxidation. So, these are the ATPase enzymes, and these are present here. Now, along with that, and the ATP and the DNA of the mitochondria is self-replicating. It can replicate by itself, but it is not as efficient as that of the nucleus of that particular cell. Now, this is a semi-permeable membrane where it allows only some of the substances to enter into the cell. So, it is partly semi-permeable membrane. It is sort of a semi-permeable membrane. So, in that way, the mitochondria is a powerhouse of a cell because electron transport chain occurs in that and it produces ATP. What are the other major reactions that take place in the mitochondria are the mitochondrial matrix is a place where most of the catabolic reactions do take place. It is a place where catabolic reactions take place. That is the mitochondria. Catabolic reactions take place. That is why whatever reduced nucleons that are formed, they can enter into the electron transport chain. Next is the cytoplasm. The cytoplasm is a basic, or it is a center structure in which all the cell organelles are present. So it is like a flowing thing that is present inside the cell, and that will help in floating all the cell organelles, making it compact, and whatever nutrients that are required. See, the cell can synthesize its own nutrients, but the nutrient has to come out, and they can directly come to the mitochondria. They have to come through the cell membrane into the cytosol. Now the cytosol will decide to send how much amount of energy is required for each cell. It has a capacity to understanding that. So from the cytosol, the things will move towards their favorable things like endoplasmic reticulum, like cellulina, we don't know about that, centriole, and then mitochondrial membrane. Golgi apparatus, you can see Golgi apparatus are the vesicles that are formed. They go out of this membrane. They will be going out of this membrane to their target tissues. So in that way, cytoplasm is a full material where there are many electrolytes are there, glucose is there, lipids are there, so many things are there. All these things are present in each state. They are present in such a way that everything is floating in the cytosol. So it is an intracellular material present inside the cell. This is enclosed by cell membrane and it is except nucleus. The fluid present in between the cell organelles is called as cytosol. So the cytoplasm is nothing but intracellular, that material plus organelles is called as a cytoplasm. So cytoplasm means except the nucleus. Removing this nucleus, what all the things that are present inside the cell is called as cytoplasm. And only that fluidy material that is present when you remove all these cell organelles along with the nucleus, the fluid thing that you can see, you call it as a cytosol. So there is difference between cytoplasm, cytosol. Cytoplasm is a wider range. Cytoplasm will include cytosol and subcellular organelles except nucleus, that you call it as a cytoplasm. The intracellular organelles except nucleus are called as cytosol. These are the intercellular things, and when you and the fluid part is called as a cytosol. Next, now we are talking so much about the cell and all that, and how the cell is retaining its circular shape like that. Why is it not falling down? Why? How is that retaining its shape? The cell retains its shape because of the presence of cytoskeleton. Now we are having this particular posture because we have bones. These bones are helping us to maintain a straight posture, or when you are flexing your hand, only at the joints the hands will bend. The hands won't bend like this or somewhere else. Only for superheroes they bend like that, but not for normal people. So our hands bend only at the joints. In no other parts of the body they do bend. So this support is given by the bone. In that way, for cell also some support should be there, or else it will get collapsed. So what gives support to the cell is called as a cytoskeleton. It is called as a cytoskeleton that includes microfilaments of actin and includes terminal web and nuclear actin. So microfilaments like actin and the nuclear actin will help in maintaining. So these are the microfilaments. Then intermediate filaments like keratin, neurofibrillins and neurolaminids can also help. Along with that, microtubules will also be there. Those will help in holding the cell during the cell cycle phase. During the cell cycle phase, the microtubules will be helping in holding a cell. So this is a bit detail about the, what is the time, 43. So this is a bit detail about the cell and cell organelles. For you, the function, you need to learn the function of individual cells a little bit in depth, mostly of that of the mitochondria. All these things you have to learn, and then tomorrow we'll see the most important part is the cell membrane. How the cell membrane, because we are concerned about the receptors that are present on the cell membrane. So we'll talk about the cell membrane in the next class. So most of you have put on comments that you want to know the list of books. So the list of the books will display it in the MIMS website within 1 or 2 days. So in within 1 or 2 days, we'll put the list of the books that you have to purchase for biochemistry, anatomy and physiology. Just wait for 1 or 2 days. It will be displayed on the website. So for today, I am closing the class. Tomorrow, not tomorrow. Yeah, tomorrow. No, tomorrow we don't have a class. We have class on next Tuesday. So Tuesday we'll once again meet.